can one of you or all of you discuss why you think retaining the Hazardous Substances and New Organisms Act 1996, the HSNO Act, as the most practical solution instead of the gene technology bill, which would become the Act, and why risk tiering inside the HSNO Act instead of risk tiering outside that prospective Gene Technology Act might be the best solution for New Zealand?
The difference between the Gene Technology Act that's being proposed, the bill that's being proposed, and HSNO is not the range of things you can do. HSNO allows you to do everything already that enable the use of a gene technology. There's a few more hoops, a few more obligations, a little bit more accountability that tracks with your use under HSNO, but it doesn't stop you. You just have to show that your product is safe and that there's a reasonable basis for wanting to put it in the environment. And sure, any regulatory process has its costs. The question is whether or not they're proportionate and appropriate. What this act does, different from HSNO, is it says, or it hopes, that suddenly there will be a new utopia of gene technology outcomes simply because nobody has to tell you what they did. And because they don't have to tell you what they did, that is going to massively reduce costs. Okay, well, personally, I'm not convinced of that. If I'm really trying to make something for a commercial purpose, I'm probably going to have pretty much the same costs anyway, because I'm going to want to control my intellectual property. I don't want that organism to be captured by anybody else during the process of production. And for all sorts of reasons, I'll be using a containment facility just for that. And the collateral will be that my organism is recorded. Now, so I don't think this is going to reduce that many costs. And it opens us up to those risks that we've discussed earlier in this interview. If you do believe, though, that HSNO is too restrictive, then the part that you could tinker with is the transition from the lab to the environment, rather than removing any pre-commercial or research-based development, removing that from some kind of use in a laboratory. If at the point in time, and this is what other countries, I mean, India explicitly says this, they restrict it to plants, but if you do it in containment and you can show that the plant you made is the one you say you made, then it's expedited for release. It meets the exemption criteria if the description conforms to an organism at low or very low risk. And that's predetermined exemption criteria. Then you don't have to go through a lot more process to go to the environment. So they have achieved a tiering that is highly compatible with our HSNO Act, where you would continue to use containment in pre-commercial or just research use. And when you want to release it, provided that it falls within some kind of defined tier or license, then you can release it. But that verification step is essential. We already know how many times somebody has said, this organism is exactly what we're telling you it is, and later it was found to not be. So we have to have reasonable standards of verification. We have to have processes that provide confidence that whoever is declaring this organism as being what they say it is can be tested and challenged and shown to be true, and then release it. And that seems to me to be the definition of proportional.